Dr. Hongzhe Li for being with us, and thank you really to Mingji, who is really the organizer, and I will hand over to him the introduction. Just want to mention, to remind you that this is part really of the activities of the USF initiative and soon USF Institute on Microbiome, obviously in connection with all our colleagues and friends from the artificial intelligence, also the USF Genomic Group, John Adams, Kenny Kim, and all the other members. So it's really merging the activities that we have at USF around this major field of research. And so Mingji, thank you so much again for organizing, and please make the introduction.
Yeah, thank you, Christian. Hey, good afternoon. Welcome, everyone. And today we are honored to have Dr. Hongzhe Li from University of Pennsylvania to give a webinar to us. And I'll do a brief introduction of Dr. Li. Dr. Li is a preeminent professor of biostatistics, epidemiology, and informatics at the Perelman School of Medicine at the University of Pennsylvania, and is vice chair of research integration, director of Center of Statistics in Big Data, and former chair of the graduate program in biostatistics at Pfam. Dr. Li has been elected as a fellow of the American Statistical Association, a fellow of the Institute of Mathematical Statistics, and a fellow of AAAS. Dr. Li served on the board of scientific counselors of the National Cancer Institute and regularly serves on various NIH study sections. And he served as chair of the section on statistics, genomics, and genetics of the ESA, and he's currently an associate editor of Biometrics, Statistica Seneca, and co-editor-in-chief of Statistics in Bioscience. Dr. Li's research focuses on developing statistical and computational methods for analysis of large-scale genomics and metagenomics data and theory of high-dimensional statistics. He has published papers in Science, Nature, Nature Genetics, Nature Methods, Nature Microbiology, Science Translational Medicine, Cell Host and Microbe, JASA, DRSS, Biometrica, Annals of Statistics, and Annals of Applied Statistics. So, very highly accomplished scholar. And again, thank you, Dr. Li, for being with us today, and it's for you.
Sure. Great. I mean, thank you very much for the introductions. Great to have a virtual seminar. I'm really happy to hear that this new initiative on microbiome at the USF. So I'm a biostatistician. So I'm going to present some of the computational work that we are working on that are related to estimating the bacterial growth dynamics and also prediction of biosynthetic gene clusters. And if you couldn't hear me well, just let me know. So we all know that in human gut, we are, yeah.
You can be speaking.
The recording has started.
Okay. There are millions, billions of, multi-billions of microorganisms.
Everybody's.
You hear me okay?
No, somebody has to be on mute.
I think for people who are not speaking, please mute. Yeah, your speaker.
Okay. All right. Good. Yeah. So in the human gut, there are billions, multi-billions of different microorganisms, and they play very important role in human health. For example, they are related to protection against pathogens, synthesis of vitamins, immune system development, and production of important metabolites such as short-chain fatty acid. Microbiome has been shown to be associated with a cancer initiation and progression. For example, a particular pathogen such as H. pylori, and a certain virus can directly lead to inflammation and lead to genomic instability and cancer. And another way that microorganism can lead to cancer is through this barrier breach, and the microorganism can leak into the human system, lead to inflammation. And the fourth mechanism is the dysbiosis of a microbial community that can also lead to immune dysregulation. So here, often people are really interested in whether you can use a microbiome to do a personalized treatment assignment or individualized treatment assignment.
Sorry to
excuse me,
we can't see the slide.
In the chat room, there are several people saying the slides. They can't see the slides, and slides do not change.
Okay, now I can.
What should I do?
Now
so
we can see the
for
slide.
those people, can you see it now?
Yes.
Yes.
Okay,
So
thank you.
let me put in maybe because of the full screen. So can you see the change of the slides?
I'm seeing slide number 3 right now.
How about this now? Microbiome metabolites?
No.
Nope.
No, I'm still seeing the same one.
Yeah.
Okay, so maybe I'll just put in the, how about here?
Now I can see slide 4.
Yeah, it's
Fine.
okay now.
Yeah.
Yeah, it's
Okay,
okay.
metagenomics. So maybe I shouldn't put in the full screen. Okay, let's try this. Yeah, so I just talk about this association between human gut microbiome and the cancer. A microbiome also plays a really important role in human immunology and immune systems, mainly through generating important small molecules, secondary metabolites, and certain metabolites such as short-chain fatty acid and the niacin. So here we are really interested in understanding how the microbial community can produce different metabolites. And so we all know now one powerful approach to study human microbiome is through the shotgun metagenomic sequencing. So the idea of shotgun metagenomic sequencing is you start with certain microbial sample, such as a fecal sample or mucosal sample, from which you can extract DNA and you perform shotgun metagenomic sequencing. So for a typical study, you will get millions of hundreds of millions, tens of millions of very short sequencing reads. Those are the raw data. And based on this short, usually 100, 150 base pair long sequencing reads. Okay. And by doing some informatics computation, you can answer the questions of who's there by performing taxonomic profiling, or what they are doing by studying the genes and pathways that those reads can be aligned to obtain the function profiling. So I'm going to show you that, in fact, this shotgun metagenomic sequencing data also provide important information about the bacteria growth dynamics. So with shotgun metagenomic data, usually people study certain features related to microbial communities. Now, common use of features include the composition of the microorganisms, or the genes, and the gene set certain pathway abundance. As I mentioned, you can just align those reads to the microbial genes and certain gene pathways. You can also study the diversity of microorganisms. You can even study the structure variation or the metagenomic single nucleotide polymorphisms. So, and those are basically some of the static features that you can obtain from the shotgun metagenomic sequencing reads. The other things we can study from the data is actually we can also obtain information about the bacteria growth rate and the dynamic interaction among the microorganisms. So a lot of computational and statistical effort are focused on how to quantify and how to model those features. So today I'm going to briefly go over with you two topics, and if I have time, I'll mention the third one. So the first topic is from the basic microbiology science question, which is how to estimate the bacteria growth dynamics based on the genome assemblies. The second topic is related to functional microbiome. I think this is where the field is moving to, is really try to understand the function of the microorganisms and also the microbial communities. So here I'm going to present a deep learning approach for predicting biosynthetic gene clusters. And if I have time, I'll talk a little bit about the translational aspect of microbiome studies, where, as I mentioned, people are really interested in whether you can do the microbiome-based heterogeneous treatment effect estimation or assign the treatment based on the microbiome configuration. So here's the ideas of studying the bacterial growth dynamics in metagenomes. So here is just a very simple cartoon illustration of a microbial community. Suppose we have 3 bacteria here, and each bacteria we have different copies of different bacteria. And if you look at those picture, some of the bacteria, if you look at closely, they are in the stage of replicating their DNA. So you see I think the red ones, you see those bacteria have a lot of new DNA get replicated as compared to the blue ones. So the idea of using the shotgun metagenomic sequencing data to study the growth dynamic is really to try to understand how bacterial DNA replicate. So for a given bacteria, the way bacteria replicate their DNA is by starting at a particular location of their genome, is called the replication origin. Then the DNA get replicated in this bidirectional way. So if this particular bacteria is actively dividing, and you tend to see more DNA in the regions that are close to the replication origin than in the terminus region. So here you can see the red one is represent a fast-growing bacterial population. The blue one is a slow growing population, and the green one is a non-growing population because you don't see the new DNAs. But this is reality, right? This is your microbial communities. But the data we observe from sequencing this community are the very short millions of very short sequencing reads. So those reads can be regarded as a sample from the genomes of those bacteria. So the computational problem we want to answer is, based on those shotgun metagenomic sequencing reads, how can we recover something like this on my right-hand side, which what you observe here are the coverage of the reads along the genome, and this lower coverage region is the terminus region. So usually you see smaller number of reads as compared to replication origin. So for the bacteria population that are actively dividing, means the ones with a faster growth dynamics, we tend to see a bigger the slope than the non-dividing population. So this is the idea of studying the bacterial growth dynamics based on the shotgun metagenomic sequencing reads. So that's the idea. So here's just another illustration of what I just said. If you look at microbial genome, usually most of the genomes are this is circular genome, and there is a replication origin. And if the bacteria are dividing, they tend to replicate their DNA in this a bidirectional way. So if you align the reads to the reference genome in the replication origin, you see more reads than the terminus region. And in fact, in 2015, Coran et al., I think, published the first paper in this in Science, where they defined the growth dynamics as the peak to trough ratio. So you have a peak coverage and you have trough coverage. And they think this ratio, so this ratio essentially corresponds to the slope that you saw here after you align the reads to the reference genome as a way to quantify. So they call the peak to trough ratio as a way to quantify the growth rate, how fast the bacteria are divided, if they are indeed divided. So this is 2015. And later on, so the limitation of this original 2015 papers is that they can only provide the estimate of growth dynamics for the bacteria with a known complete genome sequence. Because all you need to do is just align those reads to the reference genome, and you try to calculate the peak to trough ratio. And in 2016, there is another paper published in Nature Biotechnology. And later on, 2018, my group published a paper in Nature Method, where we try to quantify the growth dynamics for the genome assemblies. So the genome assemblies means, we'll talk a little bit more next. Basically, we want to quantify the growth dynamics of the species with a known complete genome sequence. We also like to quantify the dynamics for the unknown species. And the reason this is important is that in general, for most of the shotgun metagenomic sequencing studies, sometimes there are about 40% of the sequencing reads that cannot be aligned to the known bacterial genomes. So one interpretation will be those reads must be from some of the new unknown bacteria species. So this is our motivation. So here's just a very simple illustration of genome assemblies. So again, this is just a little cartoon. You have microbial communities with different bacteria, and they are at a different stage of dividing at a different rate of replications. And the data we obtained are the shotgun, very short sequencing reads. So the first step of genome assemblies is to form, so based on the overlapping of the reads, you can construct the contigs. So the contigs basically is essential, you try to assemble those reads into genome, but you cannot get the complete genome. All you can assemble is part of the genome. So based on overlapping, there's an efficient algorithm now that you can perform shotgun metagenomic assembly. So you can get thousands of this, tens of thousands of different contigs of different lengths. The second step of genome assembly is try to group those contigs into bins. So this is called the binning stage. And binning is usually done by, but essentially you clustering those contigs into groups based on some genomic features of the contigs. And so now after the binning step, you have different bins. So just think about bins is a collection of, each bin is a collection of contigs, but they are not a complete genome sequence because we only know those contigs belong to a bin, but we don't know their relative order, where they are. So they are part of the genome, but not a complete genome. And the reason I emphasize this is that, remember, so my goal here is I try to quantify the replication rate or growth dynamics of those beans, because each bean represents one species. But I don't know their replication origin. Remember the picture I showed you before? In order to quantify growth dynamics, you need to know where is the replication origin, where's the terminus region. So this is the computational problem we want to address. Is after we know the beans, and for each of the sample, essentially you just align your reads to those contigs of a given bean, and that's the data you have. Okay, so you can summarize the data like this. So in reality, suppose I know the order of those contigs. If I know the order of contigs relative to the replication origin, and for each of the samples, so here I have, for example, 5 different samples. Then after you normalize the reads count, you normalize with respect to the length of the contigs and also the GC content. You would expect to see a monotone coverage, right? For example, for the region which are further away from the replication origin, then we have smaller number of counts than the regions or the contigs that are close to the replication origin. If we know the order of those contigs, this is what you expect to observe for the samples that carry this bacteria, and the bacteria are actively divided. But when we do the genome assemblies, we don't know those orders. So what we observe is a permuted version of this matrix, right? Because we don't know the order. In addition, we also have the noise related to the sequencing. So basically, the computational problem is this. So this is what we want to know. For each of the sample, we want to figure out the right order of the contigs so that we can compute the peak coverage, ratio of peak coverage to the trough coverage. But what we observed is a permuted version plus a noise of the truth. So the question is, based on what you observed, how can you recover this true matrix? Right. So if I know that this true matrix, then for each of the sample, I can quantify the peak to trough ratio. Okay. So let me give you an example. So this is actually one of the real data set I like to show you. So this is 6 different. So this is for a given bean, so a given species. Okay. And each of the dots represent a contig. And so this is a 6 different sample of the same beans. And what you observe here are the normalized log of the coverage, log 2 of the coverage. After we normalize, we subtract the mean. And the x-axis are just the contigs, but we don't know their order. It's unordered contigs. And you have 6 samples. And we don't see any pattern, right? We don't see any pattern. So based on this data, so here's let me go back to, so here is the data we observe, right? For example, we have 5 samples, and for each of the contigs, we have the coverage. And what you can do is you just perform a principal component analysis. Basically, you treat each of the contig as observation and treat the sample as a variable, and you perform the principal component analysis, and you order those contigs based on the first principal component. After you do that, so here you don't see any trend. You order your contigs based on the first principal component. Now you see this linear trend, right? Now you see for each of the sample, you see that roughly, there's a noise, obviously. There's roughly a linear monotone trend of the coverage as a function of the ordered contig. Okay, so from this raw data, perform principal component, you can get this. Next, what you can do is you can just fit a linear regression model And from the fitted value, you can compute the peak coverage, the ratio of the peak to the trough coverage, and use this slope as a way to quantify the difference growth dynamics. So this is our algorithm. So I don't know how many statisticians are in the audience, but I'll just briefly tell you why this PCA works. Remember if we know the order of the contigs, and for each of the samples, I have n samples, I have p contigs for a given bin. If I know the order, then I know the mean should be monotone. Remember the picture? If actively dividing the coverage that are close to the replication origin, you should see a bigger coverage than the contigs that are further away from the origin. So truth is the true mean is a monotone. What we observed are the permuted version of the noise matrix. And if we perform a PCA, use a PCA, we can order the contigs. And so under this we call this a permuted monotone matrix model. And you permute the PCA, what we can show is with a fairly high probability under certain condition, this PCA can really recover and truly recover the underlying order of the contigs of a given bin. So I'll skip the details. And here again is our algorithm. Basically, I already mentioned this. So basically, you have to do this by using the data across multiple samples. That's the key. You cannot just use the data from one single sample and try to order the contigs because there are a lot of noise in there. So because for different sample, the order of the contigs are the same because we talk about the same bacteria, right? So that's the key idea, is to use the data across multiple sample. You can get a good ordering of the contigs. And after you know the order, you can just try to figure out what is the peak coverage, what is the trough coverage, and take the ratio. So this is our algorithm. We call it DEMIC. Is published in Nature Method 2018. And Scott Daniel at the Children's Hospital here in Philadelphia, he implemented this algorithm and put, now this is part of the metagenomic sequencing analysis pipeline. It made a pipeline, so you can use this algorithm if you are interested in. You can download the program. So let me just show you what additional insight we can obtain from this type of dynamic analysis on the top of typical abundance analysis. So typical microbiome data analysis, you just look at the change of the microbial abundance or relative abundance in one condition as compared to another condition. For example, in the diseased individual, in the normal gut microbial community, whether there's a shift in the composition of the microorganisms. But I think for some studies, the dynamics can also be important. So here's a study that we did a few years ago. And so this is a study of pediatric Crohn's disease, the kids with Crohn's disease. So in total, we have 90 children with active Crohn's disease, and they were treated by 2 different treatments. One is the diet therapy, and another group received the anti-TNF treatment. So at the baseline, we obtained the stool sample and the diet information, and also the measurement of inflammation measurement called FCP, fecal calprotection measurement. And after the treatment, we followed those kids for a course of 8 weeks. And at week 1, 4, and 8, we also obtained the stool samples and performed the shotgun metagenomics sequencing. In addition, we also have a control, normal children with a similar age, 26 normal children. We also performed a shotgun metagenomic sequencing. So in average, for each of sample, we have an average of 11 million pair end sequencing rate, so fairly large data. We also obtained the fecal metabolite measurement for each of the sample. So at the end of the treatment, week 8, in the anti-TNF group, there are about 50%, half of the kids responded to the treatment, as reflected by their fecal protection went below 250 threshold. And in the enterotherapy group, there are about 12, so 32%, we observe a reduction of FCP in this group. So, so there's an initial analysis where we focus on the change of the microbial community after treatment. That was published in 2015. So here's so we applied our algorithm, this DEMIC algorithm, and this algorithm were able to estimate the growth dynamics. So those are the rate of growth for 278 species, bacterial species, and 20% of the species appears in 50 or more of the samples. So here, I forgot to mention, the trick here is some of the bacteria, they only appear in some of the samples, right? So sometimes it's not a good idea to pull the data across all the samples, because if some samples, if they don't carry this bacteria, then they will not contribute your estimate of the growth dynamics. So anyway, so 278, from which we observe about 7. So we did the genome assembly, and 7 of the beans. So those could be, some could be the new species, some of the known species. Actually, most of the beans that we identified, they cannot be assigned to the known bacterial species. So 7 of them, we observe a very different growth dynamic. So that's the replication rate, very different in the samples with Crohn's disease as compared to the normal individuals. So here, just give you some idea about the completeness of the beans. Remember, I mentioned the beans are the collection of the contigs. They are not complete genome sequence, but now there's an algorithm you can estimate how complete your beans are. And some of the beans also include contamination. So those are the contigs from other species, maybe grouped into a particular bean. And again, there are some algorithms you can use to quantify the quality of the genome assemblies. But most of those 7 species, you see a high growth rate in the normal gut community than the kids with the Crohn's disease. One of them, you see a higher rate of growth in the patients, Crohn's disease patients. And so here again, I just listed 3 of them. You can see they are very different. For example, this particular bean species, then we don't know. We can only assign the first species, I think, to the order level, to the Clostridial, this level. And you can see this is estimated peak to trough ratio. In the control is much higher than the Crohn's disease gut microbiome. And similarly for the second species and the third species, we see a higher growth rate in the Crohn patients than the normal controls. But what is interesting is this. So this is at the baseline. But after treatment, remember those Crohn's disease kids, they receive treatment, either diet or anti-DNF treatment. After treatment, you see their growth rate started to go up. they start with a low growth rate and started to go up. And after 4 to 8 weeks of treatment, you see their rate now getting closer to the normal individual, normal kids. And similarly for the second species, you also see an increase in growth rate. The third species started to see go down after treatment. And here, I actually didn't even divide the patients into responder, non-responder. Just the average of all the Crohn's disease patients who received the treatment. You see a change. So this could be the treatment leads to the change of the growth dynamics, or it could be the reduction of inflammation lead to the change of the growth dynamics. That is difficult to tell which one lead to which one. But I think it's interesting to observe on the top of the abundance, you actually see the difference of the rate of bacteria replicating. So this is what our algorithm can be applied to data analysis. So this is just the first part of my talk. I introduce an algorithm, a program called DEMIC, and they are available online. And one of the key idea used in our program is to use the PCA across multiple samples to order the contigs along the genome, because we don't know the order. So and some of the theoretical analysis of that method was published in this JASA, in the Statistical Journal. Okay, so this is just the first part of my talk. The second part of my talk, as I mentioned, I think now the field is really moving to functional microbiome. It's not just look at the change of the abundance, the community shift, and people are more interested in the function. What do those microorganisms do? How they affect human health? So one way that microorganisms can affect the human health is through producing some really important secondary metabolites or small molecules. Those are natural products produced by microorganisms. So those include some of the really important antibiotics, some of the anti-cancer reagent. And so this is for human. In the soil microbiome of plant, I think they produce a lot of really, really important small molecules. So the way the microorganism produce those secondary metabolites is really, those secondary metabolites are truly coded by a cluster of genes, a small set of genes that cluster together in a genetic package along the genome. And this cluster of genes act together. They refer to as the biosynthetic gene cluster. So here I give you 4 examples. So those are the 4 different BGCs encoded by different bacteria. And you can see those BGCs, they may have different number of genes, they have different lengths, but they do share some common features. Those are for example, if you look at those BGCs, they all carry the genes with a function of transporter. So you see, they are not identical genes, but they have similar function. Right? So you just look at the color. Color represents different function of the genes. They do share some common colors or some common functions, even though they are different. Right? There's different bacteria. They call different BGC. So there's some common things that we can learn from the current known BGCs. So what is our goal? Our goal is this. It's based on now there is a database list about I think a couple hundred, let me see, list about 2,000 known BGCs from the bacterial genomes. So our goal is we want to learn from those known BGCs and try to identify all other BGCs in the bacterial genomes. Follow me, everybody. So you can see now this is indeed a prediction problem, right? Because we know a set of BGCs, like something like this, about 2,000 BGCs, and they have some common features. Then now we have about 6,000 known complete genome sequence. And my goal is to identify the rest of the BGC. We think there are still many other important BGCs in the genomes, and I want to identify them. So this is our goal. Okay. So as I mentioned, this is indeed a prediction problem. So for any prediction problem, first we need to know what is our training data set. So now there is a database called the BGC database. In its version 1.4, when we started this project, there were 1,984 known BGCs, and they can come from different bacterial genomes, and they have different lengths, different size. So what we did here is, based on those BGCs, we can convert those sequence. So those are the BGCs are the DNA sequence. You can predict the ORF and perform gene prediction, eventually convert those gene sequence into protein family domain, Pfam domains. So our the way, instead of using the DNA sequence as a predictor, we convert the DNA sequence into Pfam domains. So those BGCs, after you convert into Pfam domains, there are a total of 3,685 Pfam domains. Okay, and some are small. Some BGC only include 1 or 2 domains. I think we just removed them. We only use the BGCs with somewhere from 3 to 225 Pfam domains. So here's a histogram of the number of the Pfams of those known BGCs. So most of them, they include small number of protein domains. Some can have 250 protein domains, so varies a lot. And those BGCs belong to about 1,000 bacterial species. So those are the known, the positive cases. And for the background, for the negative controls, background, we look at 5,666 reference bacterial genomes from the NCBI database. And we also convert their sequence into a protein domain names, a sequence of protein domains. So altogether, there are 11,000 domains. And from which we construct a controls. We think that the controls are the non-BGC. We think they are non-BGCs. So here's the training is the 1,666 BGCs and 10,000 controls. So for each of the BGC or control sequence, what we have are the Pfam domain sequence. And you can think this problem is very much like a text learning problem, because you just think each of the BGC as a sequence of words, right? Those words are the Pfam protein domain names. And so this is essentially, I give you about 2,000 very important words. Those are the BGCs. And I give you a collection of non-important words. Those are the controls. And I give you all those books. So the books or articles are the protein domain sequence of the bacteria, of each of the bacteria. And I ask you to find all those important words from those articles. So this is almost like a language or text mining type of problem. So this is why we think the deep learning method might be useful. So I just want to just briefly go over with you about how we construct the deep learning. Right? So what we did is to use this long short term memory recurrent neural network. So the key of constructing a deep learning algorithm is embedding. Is you really need to create the features. Remember, our training set are the sequence of Pfam domain names, protein family domain names. So first, you can code those those Pfam word by this what they call the embedding. You can use a numerical. So what we did is about 112. Yes, you can embed those word into numerical values. So for each of the word. The second one, what we use is for some of the Pfam, we also have information called the Pfam clans. So those clans are the Pfam with a similar function. So that's another, so almost like you have two words, they have similar meaning. And the third information, the third type of information we use is a description of the Pfam. So this is almost like you give me a word, I have little description about the word. You give me two words, I have some similarity dictionary that we can use. So in our algorithm, we use all three types of information in the numerical embedding of the BGCs. And so after do the embed, at the end, each of the BGC is, sorry, not each of BGC, each of the Pfam is embedded by 1,126 dimensional numerical vector. So, and remember, each BGC is a sequence of Pfam. The longest one is 250. Then you can just run the standard LSTM recurrent neural network. Eventually, you can predict for each of the Pfam, you can predict whether they belong to BGC or not. And here another trick we used is the data augmentation ideas. So remember our training, we have about 2,000 true BGCs, and we have 10,000 controls, non-BGCs. So the sample size is still relatively small. One trick that in the deep learning field, which is very important, is this idea of data augmentation. So the data augmentation is what we use is this. So you have a BGC, right? So remember, BGC is just a sequence of protein family domain, Pfam 1 to L. So we can augment the data by creating the pseudo BGCs. So what we did is, remember, for some of the BGCs, we have the Pfam with a similar function. So we just replace, with certain small probability, we replace the true Pfam with one of the Pfam from the similarity database. Then we think this is also a BGC because they have similar function. Then you can create this pseudo cases. And this turn out help us a lot in terms of the prediction. So this is the third trick, the second trick we used in our learning. So, and this is embedding. You can see the embedding is very effective. What you observe here is, so each dot here represents one of the BGC, and this is actually not a training set. This is a validation set. What you observe here is the t-SNE plot of the embedding. And you can clearly see the BGC, non-BGC they are very well separated, even for the validation set, which are the data that we never use in building the model. So this can separate, this embedding can indeed, with the deep learning, can indeed separate the BGC, non-BGC well. And the embedding is also quite effective in separating different types of BGCs. So for BGC, we can also classify the BGCs into different types. For example, there's NRP, there's RIPP, there's different type of BGCs. And here are the result of the, so this is for the training BGC. And there's one group called other. Those are the BGCs that do not belong to any of the known classes. And some of the BGC, we don't know their classes. And you can see those different color represent different BGC class. They're all separated pretty well, except the red ones that we don't know which class they belong to. So this is just tell you a little bit about this embedding seem to be quite effective in separating the BGC, non-BGCs, and different type of BGCs. So here's the result. I'll just show you how you're going to validate your prediction result. So what we did is we leave 13 bacterial genomes out. And those 13 genomes, they are known, include 290 known BGCs. And they are never used. Those 13 genomes, bacteria never used in the training. And but we only know they include 291 known BGCs, but they may also include other BGCs that we don't know, right? Our goal is to identify other BGCs. So we also create a artificial genomes, and so we keep the BGCs, but we randomly replace the original Pfam sequence with a non-BGCs. So basically the testing data set is not a true genome, is a genome with known BGC and non-BGCs. And so here I present the prediction result on this validation data set. And I compare 3 different methods. One is called the DeepMBGC. It was published last year in Nucleic Acid Research. This one is, the second column is our DeepMBGC, we call the multi-class BGC. And the third column is our method plus the data augmentation step. So without just going into a lot of detail, I just want to point out, for example, the F1 score statistic. Usually this is quite informative statistics to evaluating the prediction result. You can see a good improvement over the DeepMBGC. So DeepMBGC, they only use the Pfam domain name embedding without using the function information, without using the similarity information. And so for most of the criteria, you see our algorithm perform better than the DeepMBGC. So this is for predicting at the Pfam level prediction. And this is at the BGC level. So Pfam is just one of the protein family is part of the BGC. Now, based on the posterior probability for each of the Pfam, you can also try to predict a sequence of Pfam, which are the BGCs. And here, but how are you going to evaluate your predicted BGC with a true BGC? So you can have different criteria. For example, if there's any overlap, you call this positive. Or at minimum, they have to have 20% of overlapping, 40%, 60%, 80% overlapping. And again, our algorithm, you see, this is a F1 scores. You always see a bigger F1 score than the published DeepMBGC algorithm. So this is just predicting whether there's a BGC, no BGC. Remember, we are also interested in predicting the class. what type of BGC that a BGC is. So here's the result of predicting the BGC class. So there's a newer version of this MIBIG database, version 1.5. They input 160 new BGCs with a known class, and this is our prediction. So multi-class prediction, because the training sample size is small, so the prediction accuracy is not as high as just predicting the BGCs. So we have about, in terms of accuracy, it's about 75% accuracy, and recall rate about 77%. So it's a little difficult to predict the BGC type. So I'm almost done. I'll just give you a brief summary. So then we apply our algorithm to try to identify all the BGCs in the known 5,666 bacterial genomes, and made a lot of predictions, 161 prediction. Now I have to admit, we didn't do a lot of post-processing. So some of the BGC could be small, which may include 2 or 3 Pfam domains, so they're small. But what we observed, for example, for the predictive BGCs, majority of them belong to this IRPP class, and called the ribosomal synthesized and post-translational modified peptide. And this is the reason we predict so many of the IRPP is those BGCs tend to have a very conserved genomic arrangement of many genes, so make it easier to predict. So I'm not going to, I think the time is almost up, I'm not going to go to detail, but at the end, my hope is this. So here are the BGCs in the species stratified by phylum. If you give me a phylum, for example, Firmicute, then I want to know within this phylum, what type of BGC this type of phylum can produce for each type of BGC. And my goal is, if you give me a microbial community, I want to quantify the abundance of different BGCs. That will provide me some ideas about what type of small molecules this microbial community may produce. And I'm going to, based on that quantification, try to link to the for example, clinical phenotypes. So this is the way we are trying to study the function of a microbial community by predicting the relative abundance of different type of BGCs and link that with clinical phenotypes. So just in summary, I present a deep learning algorithm which can perform multi-class BGC discovery, and we observe a better performance than the published DeepMBGC algorithm. In addition, the DeepMBGC can only predict BGC, no BGC. BGC, our algorithm can make a multi-class prediction. And now we have a database for all the predicted BGCs for each of the species. And I think this potentially can be useful for discovery of a new novel natural product. Ming, I think I'll just stop here. I don't think I'm going to have time to talk about the third topic. So I'll stop here, and I'll be happy to answer any questions.
Oh, thank you so much, Hongzhe.
Yeah.
That's a wonderful talk.
Thank you.
Okay, we have time for questions. Not sure.
Hey, I have a question.
Yeah.
Hey, hello, Dr. Li. So
Hi.
thanks for this very interesting topic. And my name is Charlie. And I really like your first topic. And actually, your question is how to order the contig.
Yeah.
Actually, that's a very smart way. Actually, so if you want to order it, how about you find the the direction with the biggest variation and then order it. But do you mind to remind us why this contig is so important? Because I mean, so you are trying to find the contig, how about maybe is very important related to the time series, and why this order is so important?
Yeah. So a good question. We actually tried by taking the mean, by taking the variance to order them. None of them work as well as this PCA. Now, why order the contigs are important? You see this picture. Remember, the truth is this. If I know the complete genome sequence, I can just align my reads to the genome sequence. And the more reads you observe in this replication origin, that's indicating the bacteria is actively dividing. So I really need to know where is this location, right?
Okay.
So, but when you do the genome assembly, you don't know that particular location. All you know are part of the genome, and you don't know their order.
I see.
So only when I know the order, I can roughly know where is the replication origin, where's the terminus region, because those are the two regions where I'm going to look at the reads coverage, and from which I can get the quantification of the change of the coverage. So that's why I need to order them. But the data I observed is unordered data. It's like a randomly permuted version of the ordered data. This is what I observed. And I said, how are you going to order those contigs based on this randomly perturbed matrix? And your suggestion is well taken. You are saying each of the column, just take the variance. Yeah. And you think bigger the variance may correspond to the bigger variance doesn't work well. You may think about take an average. But average doesn't work as well as a PCA either.
Okay, so I have one more question, sorry. So the next question is, I know the limitation of this microbiome study, sometimes the data may not available, but have you actually validated your method? I mean, did you using some independent data side to prove actually your method works much better than other method?
Yeah, so I didn't show you the details. So in this in our Nature method paper, we did a couple comparison with a bronze paper with this paper. So bronze paper, I just want to mention. So what they did is they order the contigs based on the data that you observe. You cannot do that because there are lots of noise. So the key is you have to order them based on the data from multiple samples. So we compare, yes, we use the synthetic data. We also use some of the real data set to compare those two different methods. I didn't show you any of the comparison in this talk, but this paper you see actually quite a lot of comparison to convince the readers that our method indeed perform better than bronze method. And you can see statistical, you can see their method won't work because they did this ordering one sample at a time. The key is you have to use multiple samples.
Okay.
Yeah.
Thank you. Yeah.
Sure.
Hi, this is Sam Mohapatra. Really enjoyed your presentation. One question I have is, once you determine the BGC for a particular disease, you think it's going to be the same for other diseases, or it's going to vary from disease to disease?
Those are good questions. The BGC at this moment, we only get to the level of predicting the BGC. Actually, we haven't really compared the disease sample and the normal sample yet. That would be something we like to do next. But that's really where we're going to do. But I don't have any result on that type of comparison. I'm hoping they will be different.
Yep. Yeah.
Yeah.
You think they will be
probably more stable than the abundance.
Yeah. Thank you.
Yeah.
Great. Any other questions? Any other questions from other people?
Hello.
Yes.
Hi.
Hi, this is Sudeep Sarkar. I'm in computer science, and I really enjoyed your talk. Thank you for presenting to this group. I learned a lot. My research is in not this area. I do deep learning for computer vision. And video processing. So some of the tools are similar. I can see that. I enjoyed it.
Yeah, this is just a very simple application of deep learning.
Yeah, they're very nice. So I was going to ask you, do you see a need for new architectures in deep learning to solve some of the core problems, or are traditional architecture enough?
So I'll tell you the limitation. One is the training size. I think is small here. The other things I actually really like to do to try if there are better algorithm is so the BGC, what we did is at this protein domain level, you can convert the DNA sequence into protein sequence, not a domain. That, I don't know, may work better in terms of predicting those BGCs. So I don't know. The deep learning, I have limited knowledge, but I'll tell you one question that puzzles me is this multi-class BGC is some of the BGCs, they belong to multiple class. So they have multiple functions. Now we only just pick one. And how are you going to use that? I don't know whether there are better algorithms in the deep learning field. Yes. So that's something I think would be really interesting for the computer scientists to have a better way to use that information.
Yeah,
so
multiple levels.
BGC, they may have multiple functions.
Yep.
But I don't know how to. So currently, we just take one in our evaluation. Yeah.
Well, thank you.
Sure.
Thank you very much. More questions?
Well,
Any questions from the clinicians, the scientists who actually work on microbiome studies? All right.
Thank you very much.
Thank you so much, Hongzhe, and
great.
Thank you so much, Dr. Hongzhe. Thank you so much.
You're welcome.
Great.
Very nice giving this presentation.
So, Hongzhe, could you send us the presentation?
Okay. Yeah, sure.
And Jerry has recorded this seminar webinar, so she will also send the link to the group.
Okay.
Yeah,
and Jerry, you will post it, all of this, you post it on the website, obviously, of Institute.
Yes, of course.
So there's a website. I'll look at your website.
Yeah, please, Jay, send the link for the website to Dr. Hongzhe.
Yeah, I'm interested because Ming mentioned this initiative to me before. I thought it's really nice to have this.
Thank you.
I look forward to reading stuff from your group. Very nice.
Thank you.
Thank you. Bye-bye.
Okay, bye-bye.
Bye.